My name is Maynard Zerone.
And today we'll be doing the Cellular Respiration project, and we'll be doing the experiment with you guys. Okay. So before the experiment started, we already labeled the germinated seeds in this tube along with the centimeters up to 6. And that would be the non-germinated tube.
So now we're going to be adding 5 drops of distilled water into 2 of the weld plates. Now we're going to be adding sodium hydroxide into the same wells that we added the distilled water into, 5 drops.
Now we're gonna grab 2 cotton swabs and add the cotton swab into each plate just to saturate it. And we're gonna be adding 2 cotton swabs, 2 dry ones into each of the germinated and non-germinated, and add the saturated cotton swabs on top of these.
Now that we've placed the 2 saturated cotton balls on top of the dry cotton balls, Nicole's going to be adding 50 milliliters of distilled water into the beaker, which is half of the beaker full. And then she's going to be adding in 2 drops of food coloring and then swirling it to make sure that the food coloring has spread throughout all the water. Now that the beaker is halfway filled, she's going to be adding in the 2 drops of food coloring and swirling it. Good job, Nicole.
So now what Maynard's gonna go ahead and do, he's gonna grab a piece of tape, grab the germinated test tube, go ahead and do it up to halfway, right around here. It's 0.5 from the actual end of the beaker. And that's just gonna go ahead and sit there. And now I'm grabbing the non-germinated one and doing the same exact thing.
Put the tape to it. Tape is not underwater. So now that we've taped the test tubes into the beaker, we're going to be taking pictures of it every 30 minutes and inserting those pictures into here. And then at the end, we're going to be inserting our graph and all our data combined together.
So now we're gonna go ahead and get started on part 2 of the experiment, and we're gonna be showing you guys that as well. So now we go ahead and we have a cup that is half full with water. It is 32 Celsius, and we're gonna go ahead and be adding the yeast and going ahead and going to be stirring it off camera. So now that the yeast is added, we're gonna go ahead and be stirring it with a pipette.
So the Durham tubes were already placed in isopropyl alcohol to be sterilized, and they're right here. And all the different chemicals that we need to prepare in phenol red were already prepared, and they're in the test tubes, and they're labeled as such. So now we're going to be taking each of the respective test tubes labeled F, and we're going to be adding in the fructose into the red test tube, into the test tube labeled F, without spilling. So now after the fructose was added into there, we're going to set the test tube aside, and we're going to do this for both test tubes labeled G and M. Now we're doing the one with glucose for the test tube labeled G. Without spilling.
So now we're going ahead and uncapping every single one of the tubes, and we're going to be going ahead and adding the DURAM tubes upside down so that the bottom part is going ahead and being up while the opening is facing down.
So now this is what the yeast solution looks like after it's been mixed and been set to yeast. So now what we're going to do is we're going to add in 2 drops of the yeast solution into each one of the test tubes. Then after we add in the 2 drops, we're going to close the test tubes and we're going to place them in dark places, and over the course of the next 12 hours we're going to observe each test tube and the color change that it persists with.
We're also going to be adding pictures at the end of this, and we're going to go ahead and explain what happened and what our findings were.